Good evening, viewer. Welcome to our Glory Globe TV. Here's the breaking news. Yes, DNA can mimic protein functions by folding into elaborate three-dimensional structures, according to a study from researchers at World Coronal Medicine and National Heart, Lung and Blood Institute, part of the National Institute of Health. In a study published June 21st in Nature, the researchers used high-resolution imaging techniques to reveal the novel and complex structure of DNA molecule they created that mimics the activity of a protein called green fluorescent protein, that is called GFP. Yes, GFP, which was derived from jellyfish, has become an important laboratory tool functioning as a fluorescent tack or beacon in cells. The findings advance the science of how DNA can be made to fold into complex shapes and will help researchers build such DNA molecules for a variety of laboratory and clinical applications. And all DNA fluorescent tag that mimics GFP, for example, would often be ideal for labeling targeting pieces of DNA in biological studies and a diagnostic test kits, and would be relatively inexpensive to make. These findings really change our understanding of what we can do with DNA, said study co-author Dr. Sami Jaffrey, Greenberg Star Professor of Pharmacology and a member of the Sandra and Edward Mayer Cancer Center at World Coronal Medicine. DNA in nature exists mostly in double-stranded, twisted ladder or helical form and serves as a relatively stable store of genetic information. All of the other complex biological processes in cells are done by other types of molecules, especially proteins. Last year, Dr. Jaffrey and colleagues reported in ACS Chemical Biology on discovering one such molecule, a single stranded DNA that folds in a way that allows it to mimic the activity of GFP. The DNA molecule, which Dr. Jaffrey dubbed lettuce for the color of its fluorescent emissions, works by binding to another small organic molecule, a potentially fluorescent fluorophore similar to the one at the heart of GFP, and squeezing it in a way that activates its ability to fluoresce. The researchers demonstrated the lettuce fluorophore combination as a fluorescent tag for the rapid detection of SARS-CoV-2, the cause of COVID-19. Dr. Jaffrey and his team discovered lettuce by making many single-stranded DNAs and screening for those with the desired fluorophore activating ability, but they did not know what structure lettuce used to acquire this ability. To determine that structure, they turned in the new study to their long-time collaborator, NHLBI's senior investigator, Dr. Adrian R. Ferre d'Amari. In the research led by Dr. Luis Pascal, a research fellow at the Dr. Ferre d'Amari's team, advanced structural imaging techniques were used, including cryo-electron microscopy, to resolve the structure of lettuce at atomic scale resolution. They found that it folds into a shape that has at its center a four-way junction of DNA of a type never seen before, enclosing the fluorophore in a way that activates it. They also observed that lettuce foldings are held together with bonds between nucleobases, the building blocks of DNA that are often referred to as the letters in the four-letter DNA alphabet. The researchers said the findings should speed the development of fluorescent DNA molecules such as lettuce for rapid diagnostic tests, as well as a host of other scientific applications in which a DNA-based fluorescent tag is desirable. Studies like these are going to be essential for the creation of a new DNA-based tools, Dr. Jaffrey said. Beyond the report, Glory Globe TV, Delhi, India.